have to have a single like skeleton of pyramid that isn't the left when you get back.
I bombed my
It's not. And when you get down, she do a sponge. She do a sponge.
She's like, and 4, 9.
No, not even intermediate. Yeah. And the sponge, especially the intermediate stuff. She does. I like Okay, Lauren, so you need to look at this intermediate girl, and you have to look at. I saw or the one that's true. They were all false. I saw this. This is the main thing.
It's giving similar to look at intermediate.
No, we should be in no, that whole thing. Like the math
Is intermediate.
the advanced probably should be 80. That will give you 5 on 5.
How does one do that?
All right, guys. We have a lot to get through today, so we'll get started. I'll take attendance. Briley. Brianna Akins. Drew. Rhea. Henry. Anna. Nick. Abby.
Here.
Hannah.
Here.
Rosa.
Here.
Matt.
Here.
Jill.
Here.
Mia. Annie. Brianna.
Here.
Brandon. Sadie.
Here.
Jessica.
Here.
Jesse. William. Marissa. Marissa. Anna. Sophia.
Here.
Aubrey.
Here.
Matthias.
Here.
Olivia.
Here.
Braden.
Here.
Amber.
Here.
Katie.
Here.
Kennedy. Ava.
Here.
Kendall.
Here.
Emily.
Here.
Lauren.
Here.
Jennifer.
Here.
And Isabel.
Here.
Okay, cool. Any questions so far on anything that we talked about on phages, bacteriophage, viruses? Okay. So today we're going to start talking about how infections are started. So how a phage, a virus, can infect a bacterial cell. So the first thing that the phage has to do is recognize its host. There's contact with the host and attachment to the host, but those are mediated by cell surface receptors. I brought this up vaguely last lecture. There are proteins that the phage will have that are specific to the actual bacterial species that can then bind to the phage. So evolution has actually allowed the phage to recognize cell surface receptors that are really important for the bacteria. So it doesn't allow the bacteria to delete the genes to evade infection by viruses. So these are, the viruses have evolved these mechanisms to go for things that are important for bacteria to make. So they're usually normally used for important functions for the bacterial cell. Does that make sense? Okay. So some ways in which phage can recognize the right host. So this is an example of a phage up here that is recognizing a Salmonella enterica bacterial cell. So there are different examples. This is one of them. Binding is highly specific. So there are various molecules that can be on the surface of bacterial cells, like LPS components. Remember, we talked about the gram-negative cells having LPS in their cell walls or outside of their cell walls. So that could be something that a phage could recognize. It could be an outer membrane protein, an OMP. You don't have to remember these names, but we talked about membranes that sit on top of the phospholipid bilayer membrane. So those can be recognized by phage. There could be transport channels. Remember, those are also proteins that allow the transport of molecules in and out of the cell. So that could be something a virus can recognize. And then there's the flagella as well, that can be recognized by the phage. And who just walked in? 2 people just walked in. Right. Yep. Okay. So, so we talked about, the cell membrane and the cell wall, and so now we're kind of putting all that into context in the context of phage binding those specific components. So there's different molecules that can serve as a phage receptor, but that binding is highly specific, so it's only going to be specific for one thing or another. So this is the recognition step and the binding step. After attachment, then the bacteriophage is able to inject its genome. So if you remember, the phage has a protein capsid where the DNA or RNA is compacted inside of it. It's really pressurized in there. And so once it makes that attachment, it's able then to inject that DNA into the bacterial cell. So they only do this through the cell envelope. So they have to actually get through the cell envelope and inject their DNA. And there's 3 steps essentially that allow you to know that the phage has infected the DNA or injected its genome into the bacteria. So the phage attaches, and then you have this sheath. This is essentially the long neck of the virus. The sheath has to contract. It literally squeezes down on itself, and it brings the head, the capsid head, near the actual surface of the bacterial cell, and it allows it to inject its DNA. So the sheath contraction is really important, and then the viral DNA can enter the bacteria. Because this DNA is really, jam-packed in there, when the sheath contracts, it actually allows all that pressure to just be released and the DNA gets ejected. And then when that capsid head is now empty, it stays on the outside and attached to the cell. The DNA is now inside, but now it's termed a ghost phage capsid. So you can see here a really cool EM image where this one still has the DNA in there. This one, it's injected its DNA because it's called a ghost phage, essentially. Does that make sense? Okay. Oh, here's a video illustrating what I just told you. You can actually see the volume. There is volume. I guess there's not volume. Okay, so that's a bacterial cell, the phage. Those tail fibers are what initiates the contact and attachment. Once you have attachment, then the sheath will contract, and the DNA, sure, it's still attaching, starting to contract, and it spools that DNA the viral DNA into the cell. So now the viral genome is inside of the bacterium. So it has to make a choice. Some viruses, they do more than just end up getting in there and they replicate. Some of them end up stabilizing their genomes in the host. And so this essentially is called a prophage. So it's another term that you guys will have to know. A prophage is when the viral genome gets stabilized into the host and it circularizes to form a stable plasmid, essentially. Others just integrate themselves into the bacterial genome. But this step, when it's just stable inside of the bacterial cell, it's called a prophage. And so then the phage can undergo 3 different life cycles: the lytic cycle, lysogenic cycle, and a slow release cycle. So the lytic cycle essentially means that as soon as the DNA gets in, it's going to exploit the bacterial cell's machinery. It's going to make more progeny phage. It busts it open, it destroys all the cells around it, and it makes more phage. So it's just going to kill bacterial cells, and it's going to use the cell machinery to make more progeny phage, replicate, reproduce. So this is the lytic cycle. Does that make sense? The lysogenic cycle essentially is when the virus inserts its DNA, it stabilizes in the host cell, so we call that a sleeper cell, also the prophage. And so then it realizes that the bacterial cell is actually a mansion with nutrients, and it kind of just wants to hang out and stay. So the virus doesn't really have to do the work of replicating itself, because the bacterial cell is replicating itself and the virus gets replicated with it. So it's kind of like a sleeper cell. It's just hanging out there. And so it's just allowing the bacterial cell to do its thing to replicate naturally, and the virus is replicating within it. In the lysogenic cycle, when a phage is in the lysogenic cycle, it can switch to the lytic cycle if the bacteria ends up getting into an environment where nutrients get scarce, or there's DNA damage. The phage then has to get out, bust the cell open, switches to the lytic cycle. And then there's the slow release cycle, which is pretty much considered to be this persistent phage infection where the bacterial cell is, or the cell is just kept just barely alive. It's kind of like a zombie cell that's continuously spewing out phage, but the phage doesn't necessarily kill the bacteria. It just keeps the viral factory alive. So it's not killing the bacteria. The bacteria is kind of like a zombie cell. The phage is still able to, utilize its cell machinery to grow, but it's, kind of hanging out and doing its thing without killing the cell for its own advantage. So the bacterial cell is still able to grow, but more slowly than an uninfected cell. So it's still, has its machinery, it can still grow, but the virus is kind of taking up all of its cellular machinery to stay alive, so it's growing more slowly. So it's a slow release cycle. It's persistent. It's like a persistent infection of the bacteria. Questions? Yeah.
Out of the three of like the cycles, which like would be like the most common?
It really depends on the type of virus, but I want to say that they can actually switch back and forth between things. So if it's in the lysogenic cycle, it can switch to lytic. I guess a lot of bacteria will end up killing, or a lot of viruses will end up killing bacteria. So maybe the lytic cycle, the killing, is more common, especially since that's the one that they go to when nutrients deplete or when they're in survival mode. So yeah, but they can switch. The lytic cycle is kind of the end all be all last resort when they really need to replicate, kill the cell, which is essentially the goal of the virus in a way where it's essentially killing the bacterial cell. That's why it's a bacteriophage. Like, why would it infect it if it's not going to get something out of it, right? Yeah, good question. Any other questions? Okay. So now we'll talk a little bit more about them, in each one in detail. So the lytic cycle, it all starts with the host recognition and attachment. Then you have the genome entering, it gains access to the cell's machinery. And then once it is able to, so here we have the phage DNA in purple, gets into the cell, it circularizes, it's a prophage at this point, and then it starts to use the bacterial cell's machinery to now produce some of the capsid proteins that are going to end up forming into the capsid head. And then it's also going to replicate its own phage DNA while making those capsid heads. The DNA is then going to be packaged inside of the capsid heads. So it's just literally replicated itself. Then it's going to lyse the bacterial cell, which means it's going to burst it open. It's going to kill the bacterial cell. And then these progeny phages are then released. So now you've made a lot more phage that can then go on and infect other bacterial cells in the area. So exit and transmission is the last step. It reaches new host cells to infect. So the lysogenic pathway, essentially they have this alternative pathway to maintain their genome as a prophage, and so in the cell. So this is where the phage genome is stabilized in the bacterial host as this prophage. So it's also called temperate phage because you're in this temperate, temperate, I guess, like the word I'm looking for. This temperate zone or cycle where it's maintaining its genome as this prophage. So the phage essentially lysogenizes the host, which is why it's called the lysogenic cycle, which means that they are just quiescent as the prophage. And then one of the advantages to being a prophage inside of the bacterial cell is that they can replicate and become inherited along with the host chromosome each generation. So it's a sleeper cell, essentially, the prophages, that remains inactive, but they're reproducing just fine. So until the survival of the bacterial cell is threatened, then it can trigger that switch to the lytic cycle. So some of those things are UV exposure, deprivation of nutrients, other triggers. Phage lambda, I'm not sure if you guys have heard of this one before, but it's a very well-studied phage that infects E. coli. It's the classic example of this temperate phage that undergoes this lysogenic life cycle. So here we have the host cell, the host genome. Remember, bacteria also has its own DNA in blue. the phage attaches to the host, inserts its DNA, circularizes, and then it can go to the lysogenic cycle, where it essentially integrates the DNA into the bacterial DNA. And so now it's integrated, and then it actually can then start reproducing with the bacterial host genome. And so as you're making more bacteria, you're making more phage. Now, if there's some sort of stress that occurs in the environment, that will induce excision of the phage DNA, so it will remove it from the ends of the bacterial DNA, and then it can enter the lytic cycle. Does that make sense? Remember, the decision between the two cycles is really dictated by any environmental cues, anything that could threaten the host cell. So it's not just threatening the virus, it could be something that's threatening the bacterial cell. Then the virus knows that that's happening, and so it's got to get out while it can before it gets killed itself. Okay, and so the slow release cycle of a persistent phage. The phage basically produces more phage particles without lysing the cell. And so the host can still continue to reproduce, but it's more slowly than cells that are uninfected with phage. But the resources are still being used to make the phage, so that's why, you know, the host is much slower at reproducing itself, because all of those resources are being used by the phage. So you have a phage that inserts its DNA. This time it's a filamentous phage in this picture. And so it circularizes. And then, you know, you don't have to know that it forms a double-stranded circle for this example in particular. But essentially, the phage can then, you know, replicate, make more of itself, start making some of those proteins, and then it assembles and it's able to exit the cell without lysing the cell and letting it burst. So it's essentially reproducing and it can exit, but the bacterial cell isn't killed. It can still reproduce, just a lot more slowly because it's infected. So it's a slow release cycle. Basically, the slow is referring to the bacteria growing slower. The release is the phage is being released from the bacteria without killing the cell. So it's persistent. Bless you. All right, any questions on any of these? Yeah.
When, when the cell is like replicating with the phage inside of it, does it ever like physically modify like the bacterial cells? Like will the bacterial cell like change over time?
It's possible. Yeah, it definitely is possible for that. Yeah. Any other questions? All right. That's the end of this chapter. And so next we are going to switch gears and talk about transcription and translation. So the cells, they have these tiny machines essentially that make RNA, an RNA message from DNA, and then they decode that RNA to make a protein. And so that's transcription and translation. And so we're going to talk about all of the different components of the things that make up the processes of transcription and translation. We'll go through more sections, but for this part of the lecture, I'm just going to have these three. So just kind of introducing, you know, some of the terminology you guys will have to know. We have RNA polymerases, and these sigma factors that essentially tell the RNA polymerases where to go and what to do. We'll talk about how transcription works, the genetic code, which you should know from Bio 101 or 131. We'll talk about ribosomes, tRNAs, and then translation, actually making mRNA or making protein from mRNA. Okay. So the cell can access its genomic DNA through two ways. So now we're talking about bacteria. So we're going to forget about phage for a little bit. We're going to talk about bacteria, just to orient you guys. In transcription, you are reading a DNA template to make an mRNA copy, a messenger RNA. And in translation, you're decoding the RNA to then assemble proteins. So in transcription, we have an enzyme, RNA polymerase, that can read the DNA template to make that copy. And then in translation, there's this massive RNA protein machine called a ribosome that can synthesize polypeptide sequences from amino acids that then make a protein. And then it doesn't stop there. After translation, there has to be some proper folding and placement at the correct site. So there's more post-translational mechanisms in store that we'll talk about more next time. Okay. But let's start with transcription, and you're going to have to really understand the RNA polymerase and the sigma factors here. The RNA polymerase is this complex enzyme that carries out transcription by making RNA copies, those are called transcripts, from a DNA template. In bacteria, the RNA polymerase is made up of a couple of things. And so we call the entire RNA polymerase a holoenzyme, but that consists of a core polymerase, which essentially has two alpha structures and two beta structures, alpha 1, alpha 2, beta, and beta prime. These are required for initiation and elongation, and I'll talk about this more. And then the RNA polymerase holoenzyme also has a second component called the sigma factor, which is just required for initiation but not for elongation. So the sigma factor is actually really, really important. They tell essentially the entire RNA polymerase holoenzyme which gene set to transcribe given the set of environmental conditions in that environment that they're in. So if a bacteria is somewhere and there's low nutrients, then the sigma factor can tell the polymerase to make more flagella, for instance, to then get out of there and try to forage for some other nutrients. So it provides the specificity of which gene sets they have to turn on under a given condition. So I've told you before that bacteria are really ubiquitous and they're able to adapt to different environments and they can turn genes on and off. Well, now we're learning why and what contributes to that information. It's that sigma factor that is really required for that initiation of the entire process. So these are the 5 subunits that make up the entire holoenzyme. You have over here, just look on the left, you have your alpha subunits, and then you have your alpha subunits are right here, your alpha 2. These are your sigma, this is your sigma subunit. You don't really need to know that there's 4 parts to it, you just have to know that this is the sigma factor. And so this is the DNA, for instance. It goes through it, and so this is where it becomes transcribed. The beta prime, you guys should know this. So the beta prime here in this teal blue is where the DNA is really being entered through. So this is the catalytic site, the enzymatic site for RNA synthesis. So if you kind of turn this 110 degrees on its side, you can see the alpha 1 because it's hiding back there. You have the alpha 2, and then you have your regular beta subunit and your beta prime subunit. So from this angle, you'll have the DNA coming in from this part where the beta prime is. So you guys should know that the catalytic site for RNA synthesis is in the beta prime. So the sigma factor in red actually loads onto the polymerase where you see this dashed line in black. And so it will load right onto there, onto the catalytic site, kind of will clasp the DNA channel in yellow in. It'll just, kind of cap it off. And so that's what's needed for it to recognize the promoter that is going to start the process of transcription. Questions on that so far? Okay. So the sigma factor first will allow, it will have to bind the polymerase in that dashed line area, and then it helps the polymerase detect the specific DNA sequence where it's gonna actually start transcription. That specific DNA sequence is called the promoter. So the promoter signals the beginning of an open reading frame, which is where the genes are that are going to be transcribed. And a single bacterial species can make many different sigma factors. Can anybody tell me why a bacteria might want to make more than one sigma factor? See who is paying attention. Yeah.
Because there's going to be like different conditions, like it's not always the same condition.
Yeah, exactly, because they are going to be, there's going to be different environmental factors that might cause stress, or, they need to have many sigma factors on hand to then start turning genes on that need to be turned on. And so every cell has a housekeeping sigma factor, which is a gene that has to be there. It's going to be there regardless. E. coli, sigma 70. But the important thing here is that when you have, transcription, so here are two promoters that essentially will code for the start of different genes. So we'll look at this one in yellow. This is just one gene, which means that the RNA is going to be monocistronic because there's only one gene that's going to be transcribed. The promoter is the start site where transcription happens. And so what you guys should know is that the sigma factor will bind the polymerase on top of the DNA, and it will allow this polymerase to then find the promoter and sit on the promoter and then do transcription of this gene. What's cool about bacteria is that you could have a promoter that drives transcription of a gene set. So more than one gene that are probably involved in a related function. So that RNA transcript that's made is called polycistronic. So you would need another sigma factor to bind to the RNA polymerase to make that holoenzyme. It would then guide the entire RNA polymerase to bind to the promoter, and then it can start transcribing that DNA into that RNA transcript. Does that make sense? Any questions on that? Okay, so again, the cell constantly has to adjust its gene expression profile in order to survive, and sigma factors help the cell readjust that physiology. So different gene sets have to be turned on or off, and this is largely done by different sigma factors to deal with these different conditions. Remember, different gene sets can control changing conditions like heat stress, starvation, biofilm formation, flagella synthesis. There's so many things, there's so many sigma factors that can control different genes for different things. Okay, so quick concept check. The sigma factor is directly required for what? Yeah. A, just initiation. Okay, does that make sense? Also, I noticed that I actually reused some of these concept check questions on the exam, and I noticed that a lot of people still got them wrong when I've given you the questions before. You should have known the answers because we talk about it in class. If you weren't here, it's recorded. You could have asked your friends. So those are easy points that you guys should not be missing. So keep that in mind next time when you're studying, that I will be picking from these questions and reusing them on exams. That was just a real bummer to see. 3 points there. Okay. Now that I've described some of the parts that make this transcription process move, well, let's talk about the actual 3 phases. So you have initiation, elongation, and termination. Now, I want to just preface this with, you're gonna see these things again in translation. You're gonna see initiation, elongation, termination. So from now, start thinking about it in terms of compartmentalizing the two. Right now, we're just talking about transcription. There's going to be similar terms, so it's easy to get confused. But just, recognize that we're going to talk about transcription now. We'll do translation on Wednesday, so you have enough time in between then to start really familiarizing yourself with transcription so that you don't get confused when I start bringing up terms like initiation, elongation, termination on Wednesday for translation. Okay? So initiation, we kind of already covered it. The RNA polymerase, the whole enzyme has to bind to the promoter. You now understand the backstory about the sigma factor and how that brings the RNA polymerase to the promoter. But when it does that, it essentially, remember, we're working with DNA. DNA is, a helix, and it's going to be double-stranded. It's a helix. And so it's going to melt the helix, and it's going to start to synthesize the first RNA nucleotide. So it will melt open the DNA, and it'll place that first nucleotide down. In elongation, the RNA chain is then extended using the 3' hydroxyl group, which I'll show you. You're sequentially adding these RNAs, ribonucleic acids, to the 3' end of a growing chain. And then there's termination, where there are sequences at the end of a gene that trigger the release of the polymerase as well as the entire RNA transcript. So it detaches from, the RNA polymerase will detach from the DNA, the transcript is done, and so that can also be detached. And there are certain sequences at the end of the gene that allow the RNA polymerase to know that we're done here, you can remove yourself from the DNA, the transcript has been made. So that's just a quick intro of what those are, but Let's look at this step by step. So here we have an RNA polymerase, the holoenzyme, that will find the promoter sequence, and look at that. That sigma factor is right there, leading it. Then you have, it finds the promoter sequence and it closes around it, it binds to it. And then once you get binding to the promoter sequence, we then have this conformation change from a closed complex to an open complex, essentially where the DNA is then being unwinded. You have here, when the DNA is being unwinded, you start to have nucleotides being added to the 3' end. So remember, DNA has a 5' to 3' complex. And so if the RNA transcript is being made on this bottom strand, it's being made on this top strand, then the RNA is going to be in the 5' to 3' end because the DNA is in the 3' to 5' end. Does that make sense? You guys don't have to know the -35 and -10. That's just the positioning of where the sigma factor usually is. But what you should know is that, you have the sigma factor, it allows binding of the polymerase, and then you start to unwind the DNA. You start making the mRNA transcript, so it's going to be added to the 3' end. So you have these RNTPs, these ribonucleotides, the things that make, it's ribonucleic triphosphates. So these are the peptides or the amino acids that are going to be added to the RNA. And then the sigma factor actually hangs around until about 9 base pairs are added, until it kind of makes sure, like, okay, I did my job, and then it just kind of dissociates. The reason for this more open type of complex is because it allows basically the polymerase to bind really tightly to the DNA, and so transcription can occur. So in a lot of these things, for something to actually start, there's got to be a conformational change. So you'll hear that a lot. So that's initiation. Now elongation is the sequential addition of those ribonucleotides from those RNTPs. So you just essentially, RNA polymerase without the sigma factor, because it's removed now, and elongation. Remember, sigma factor is only required for initiation. So when you get to the elongation step, you don't necessarily have to keep adding, or you don't have to keep the sigma factor. It's done its job, it can leave. So now the RNA polymerase can continue to move along that template strand and synthesize more RNA. It actually occurs at a very, very, very fast rate, about 45 bases per second. And so as a result, you start to get these positive supercoils building up upstream. We call those a transcription bubble. And so you need some other enzymes called DNA topoisomerases that will relieve the supercoils upstream of the transcription bubble. So they essentially will relax the supercoiled molecules. So because there's this very quick, addition of all these nucleotides, the DNA upstream is going to start kind of getting wound and supercoiled. And so there's some other enzymes, the DNA topoisomerases, that end up helping to, unwind those knots so that when the RNA polymerase gets there, it can actually do it. So think of like actually having, trying to run something on a shoelace that's knotted. It's going to get stuck. So there's got to be enzymes to unravel that so that the RNA polymerase can continue along the length of the DNA. There will be pictures of that somewhere at some point. Make it make more sense. So elongation is just sequentially adding those ribonucleotides. You should know that the sigma factor is no longer there. And then you have transcription termination. So all bacterial genes use one of two known termination signals. There's a row-dependent and row-independent. So the row-dependent termination relies on a protein called Rho and a strong pause site at the very end of the mRNA transcript, the 3' end. Row-independent requires a guanine cytosine-rich region, lots of GCs in the RNA, as well as 4 to 8 consecutive uridine residues. So remember, the nucleotides in DNA are A, T, C, and G, adenine, thymine, guanine, cytosine. But in RNA, instead of thymine, you have uracil, and so that's why you have these consecutive uridine residues. So it doesn't require a row. And I'll explain this on this slide. So here on the left, you have row dependent. So to orient you, the DNA is right here. This is the RNA polymerase. And so you have this pause site right here, okay? And you have this row protein right here. And so this is the DNA and this is the RNA. And so what happens is the row that's shown in yellow is then able to bind to the RNA transcript, and then it kind of pulls on it, and it starts to pull itself closer and closer until it actually gets to the RNA polymerase. And once the row protein contacts the RNA polymerase, it causes termination. So it has to touch it, and then it can essentially rip the DNA and the RNA from each other, and it tells it that we're done with transcription. In the row independent type, at the end of one of those transcripts, there's going to be a rich region of GC. So this is one of those loops that we talked about. So there's a bunch of GCs. And so that actually allows this hairpin loop to form just because of the bonding between G's and C's. It allows, it just causes this hairpin type of loop. And so then that, will eventually block the RNA polymerase. It can't go anywhere. It's encountered this hairpin loop. So if the RNA polymerase is here, it's on top of this hairpin loop, and so it's unable to move. So contact between the hairpin and this other protein that's involved, NUS-A, you don't need to know that, then that causes termination. So this is row independent because it doesn't require the row protein. Okay. Any questions on this? That was a lot. We went from viruses to bacterial transcription. Okay, we will stop here and we'll finish off on Wednesday.
Burn on my back.
I'll email you as well, Bob.
Yeah. And then I think I might be starting.
Yeah, we're trying to look at Jordan.
That makes sense. You are Nick. That checked out.
I'll just write it here since I have this. You don't have to email me.
Thank you.
Hi, I want to so on the 28th. my doctor just to make sure. I haven't skipped that class.
Okay,
I
yeah.
also have another doctor's appointment. I'm gonna make sure on the time and I don't know if I mix or not.
We can make your exams, you're letting me know in advance, that's totally fine with me. Yeah, so just let me know what works for you for the exam, and then we can work around that. Yeah, how'd your MRI go?
Not very good.
Really?
Yeah, he wanted me to actually get surgery today, and he said that he could wait until after my regional meet for equestrian, because if he did my surgery today, I wouldn't be able to compete.
So you're still gonna compete on your?
Yeah.
Oh my god.
I competed last year when I broke my foot the night before.
Oh my god. That's
Yeah.
crazy. Well, good luck. I hope. Oh my
Thank
god.
you.
I can't believe you're gonna require surgery now. That's insane.
Yeah, it's a little scary.
Yeah.
Thank you.
Yep. Okay.
Still good to come to
2 o'clock. Yep. Perfect. Yep.
I'll be there. Thank you.
Yep.